Good morning and good afternoon, everyone. Welcome to the webinar, Mining Somatic Variants in Cancer Exomes Utilizing Bina AAiM. Your presenters today will be Anoop Graywal, that's me, I'm Product Marketing Manager, and I support the AIM module. I'll be giving a brief introduction, and that'll be followed by a live product demo by our Senior Scientist, Aparna Chibber. We'll follow that with a Q&A session. So a few guidelines if you'd like to ask some questions. You'll see a green bar at the top of your window indicating that you're seeing the presenter's desktop. If you just hover over that with your mouse, that'll bring down the control panel. If you click on the Q&A button, that'll open up the chat box. And go ahead and type questions in that Q&A chat box, choosing to send to all panelists. That means we'll be able to see it and answer your questions. So this is the Q&A window in which you would ask the question. All right, let's get started. So AIM stands for Annotation and Analytics Intelligence Module. In the GMS or Genomic Management Solution Framework, this is the component that performs the tertiary analysis of the data. The input into the Bina AAiM module will be VCF files. So VCF are the product of secondary analysis. You may have generated these files using the Bina AAiM platform or your own proprietary secondary analysis platform, or you may have received VCF files from your sequencing core or service. Regardless, the VCF files are going to contain millions of variants per sample if you've done a whole genome analysis, or tens of thousands of variants per sample if you've done a whole exome. And the challenge at the next stage, then, is to narrow down to the handful of variants that are either going to be disease-causing or predisposing to disease. Now, that's what the Bina AAiM module is for, and to this end, we're leveraging a lot of well-regarded databases out there that represent the current knowledge in the field. These contain information about variants, genes, and genomic regions. On top of that, we've integrated a number of tools and visualizations which are going to help you in your interpretation as you narrow down to a handful of variants that you can then analyze one by one. Now, we have a number of features as part of the product that make it pretty powerful. Overarching is the breadth of the annotations in AIM. We have 140 plus different annotations culled from over 20 different databases. One of the things I think you'll notice when you see Aparna give her demo is the speed with which you can apply filtering and querying and then change your filtering threshold and see the results change relatively rapidly in just a matter of seconds. Another place where we pay a lot of attention is to have a very easy-to-use interface, one that requires no bioinformatics expertise. And finally, we've tried to make the filters as customizable as possible. So for instance, you'll notice in the demo that there'll be some relatively easy out-of-the-box filters so that you can hit the ground running, but you can customize the associated thresholds, and as well, it's scalable. You can apply the filters to a single sample, to many samples, and then you can save the filter setup so that you can apply it to future samples. Those 140 plus annotations I was talking about map to a number of different categories. So there are a subset of those that will give you information about genomic context. For instance, where is that variant located in the genome? Is it, for instance, in the protein coding portion of a gene? Is it in the transcribed portion of a gene? Or is it in extragenic sequence? This type of annotation and other related annotation can be found in some public databases, RefSeq and Ensemble, as well as a few commercial databases that we've integrated, such as Transfac and Proteome. Another useful piece of information to reference against is population-level frequency. You'll probably have a very specific idea of the likely frequency at which your variant is present in populations. And so to this end, we've integrated frequencies from the Exomes sequencing project, from dbSNP, and from phase 3 of the 1,000 Genomes project. Just to drill in a little bit more, the Exome sequencing project, for instance, also contains frequency at an ethnicity level if you want to filter accordingly. If you happen to be looking at the non-synonymous variant set, one of the important questions is what is the predicted functional impact where there are amino acid changes or changes to the splice junctions. And to this end, we have annotations such as mutation tester, polyphen2, SIFT, LRT, and SNP-F. This is going to allow you to, for instance, filter for mutations that are likely damaging, or select for mutations that represent frame shifts or other types of codon changes. So here are the general categories more broadly that they fall in. I've highlighted a few, but for instance, with respect to clinical annotations, there are a number of sources that allow you to ask the question about whether the variant or gene has been previously associated with disease or clinical phenotypes. And with respect to regulation, whether some non-coding variants, for instance, fall into a regulatory element. More specifically, here are the databases representing each of those categories. And I didn't delve into clinical annotations, but for instance, there's some very useful data sources there that you can filter against. The Clinvar effort, for instance, the Clinvar annotations, that is, Cosmic, the Cancer Genome Atlas. You can both filter on these annotations, and when you export your results, carry over the annotations associated with that. So at this point, I'm going to pass over the presentation to my colleague, Senior Scientist Dr. Aparna Chiver.
Good morning, everyone. So I'm going to be going through a demo of the product. And for today's demo, we're going to be using Ames to study exome sequencing data from the National Cancer Institute's panel of 60 cell lines. And during this demonstration, we're going to be using the panel to highlight the databases and visualization tools available in Ames to facilitate variant interpretation from next-generation sequencing data in oncology research. So this is just a quick overview of the panel. There's a few different cancer types or sites represented in the panel. So we're going to be showing you exome sequencing data from all of these cell lines combined in the product. VCF, or variant call format files, for each of these cell lines have already been uploaded to AIM and processed. Just to explain what that processing step does, since you're not going to see it during the demonstration, during processing, uploaded variants are annotated against REST-seq gene models, and that step is using SNP-F, as well as the 20-plus databases that Anu walked you through in the introduction. And once that pre-processing step is complete, the samples are available and ready for querying in AIM. I'm going to switch over to the product now to show you the demonstration. So here you can see some of the projects that I've uploaded into Ames. I'm going to select a project I've created for this cell line panel. Here the 60 exomes have been combined together, and we've also assigned each cell line to a group. And these groups are based on the original source tissue for each cell line. So this is that pie chart that I showed you on the slide a couple seconds ago. Each group is based on that original tissue source. So we're ready now to start querying. This is the primary query interface of Ames. This view is showing you right now for a single cell line, and it happens to be a renal carcinoma cell line. So this is showing you for a single cell line all the variants in this sample. The table shows you below a summary for each variant of the gene the variant falls in, the predicted functional effect, again from SNPs, the position in the genome. This is also a link to the UCSC genome browser, the genotype for the variant, and a quick summary of the databases that the variant falls in. Now we can start adding some filters to these variants to try and narrow down on potential variants of interest. So on the left pane here, we have some predefined or Bina-defined filters. And these are available for filtering on features such as quality, allele frequency, functional effect, location in the genome, disease association, pharmacogenomic properties, and so on. Let's start by filtering down only for high-quality variants. If I open up this filter, you'll see here the query definition. Any of these default values that we've defined here can be changed by the user. You can modify these as needed. And the advanced view shows for any filter the formal query definition for the filter. Again, this view can also be customized. So if I apply this filter now, this step is going to remove any variants that don't meet the criteria specified in that filter. Next, let's select for variants, novel or very rare variants that are not present in any of the population databases that Anoop mentioned, or that are present in those databases at very low frequency. We can also select for variants most likely to cause a change in protein function using the damaging filter. And finally, let's also go in and filter for variants that have been previously identified, either previously identified as somatic variants or identified in genes that have been identified in cancer samples previously. And I'm going to remove COSMIC from this list of databases just because variants from these cell lines have already been deposited in COSMIC. Now we can save the set of filters that we've created to use at a later date as well. And also get a graphical overview of this set of variants that we filtered down to. So now that we've come down to this 452 variants, if we want a quick graphical overview of the properties of the variants that we filtered down to, we can use the dashboard view to get a quick overview. And here again, I'm showing you some predefined graphics. Just as with the filters, you can create custom visualizations and graphs displaying any of the information that you'd like for the variants in your samples. Now, so far we've been looking at only one sample. We have 60 samples in this project, so we'd likely like to look at these holistically. And we can apply the filter set that we've created to all samples in the project using the multi-sample views. So I'm going to switch to the multi-sample view. Here, this set of 4 filters is being applied to all 60 exomes. And we get a quick summary here for each sample of the number of variants after filtering. We can also focus on a targeted set of genes of interest using the gene panel function. And I'm going to switch to the gene panel view here. I'm going to select a gene panel that I've previously uploaded to the system, but again, these are the gene panel function is completely flexible for the user, so you can upload any panel of genes that you're interested in studying. In this view, we're looking at, for each gene in this panel, the number of samples in each group that have a variant in the gene. So for example, there's one breast cancer cell line with a variant in HRAS. The summary graphic on the right side shows the same information as a graphical overview. So here, each gray rectangle represents one sample in the project. If the rectangle is colored in, that means there's a variant in that sample for the gene. And the coloring here is coded by group. So for example, all the breast cancer samples are colored brown, the leukemia samples are colored blue, and so on. So the summary provides a quick way to get an overview of the mutation profile in this project. We can quickly sort this table here just to pull out genes of interest to the top of the table. Now, of particular interest when studying cancer are mutations that are clinically actionable. So to pull out potentially clinically actionable mutations, go back and create a custom filter that selects for any variant that's been previously reported in a pharmacogenomic database. So here I'm going to create a custom filter. I'm going to select the pharmacogenomic mutation database. Again, you can see that any of the databases here are available to create a custom filter. And I'll just include all the variants for now in this database. To get more detailed information about particular variant annotations, we can use the detailed view. And pull in some annotations, again, from any of the databases available. I'll pull in some annotations from the pharmacogenomic mutation database, since that's the one we're focused on here. Now let's apply this new filter set that we've created, including the pharmacogenomic filter, to all samples in the project. And we'll switch to the gene panel view and just focus in on genes that have been previously reported to be clinically actionable. Here again, we can use the mutation summary to quickly pull out genes of interest. So for example, BRAF here looks like it's mutated in almost every melanoma sample. We might want to study it in a little more detail. So I'll click on BRAF. Now in this view, we're looking at a summary of variants just in BRAF. In this plot on the top, the x-axis represents the amino acid position for the gene from beginning to end. And the y-axis represents the number of samples in this project with the mutation at any given amino acid position. And colored rectangles here represent the position of protein domains. So using this view, we can very quickly see that mutations at position 600 in the tyrosine kinase domain occur in 11 samples in this project, and this variant might be of interest to study further. And the table below shows us more detailed information about this variant and the samples that it's found in. And this mutation, incidentally, this is a known mutation that we pulled up as a new sample. The mutation is known to be associated with an increased sensitivity to proteasome inhibitors. This is a good time to open up to questions from listeners. Again, you can use the Q&A under the green bar at the top. If you go to options, you'll see a Q&A section, and if you click on it, you can type in questions there. So we had a question early on while we were going over the databases about EXAC. We will be adding EXAC, or working on adding EXAC databases to our set of population frequency databases. There's a question about the limit of detection for somatic mutations in our software. Our RAVE software, that's secondary analysis, does have a very detailed somatic detection pipeline. I think there will be a follow-up webinar later in the summer covering that, and that would be a good place to learn more about our mutation detection pipelines and tools. There's a question about tumor normal pairs. So what you're looking at here, in this example, these were cell lines. This is exome sequencing, so there was no matched normal. If you have matched normal pairs, we'd suggest you use tools specifically designed for looking at tumor normal pairs together. And again, our RAVE software has a great set of tools for doing tumor normal calling, variant calling. There's a question about getting common variants. So I'm going to guess this means common in terms of population frequency, and yes, you can indeed. I showed an example of a novel variant filter. You can get variants that are common in population databases. There is a question about clustering analysis or principal component analysis on these samples. That's not currently something that we have in the product, but we are building out our analytic capabilities greatly. So that is something that we may have in the future. But currently, yes, the data would have to be downloaded and analyzed separately in another tool. There's a question about uploading BCS with multiple samples. You can do that. So you can upload a multi-sample VCS, or you can upload a group of VCS separately at the same time. Either option is available.
While waiting for another question, I just wanted to remind everybody we will make the recording of this WebEx available, and we'll send you an email about that.
There's a question about deployment. I didn't mention that. So this product is completely on the cloud. So the product is only available on the cloud. It's not software that you can download and install locally. And if you have questions about costs or further, you want further details about how you can try this software at your own institution, I'd encourage you to contact, go to the websites that are on the screen right now. There'll be contact information there. If you go to the test drive page, there'll be a form to fill out, and someone from Bina will contact you again shortly. There's a question about variant colors that we support. So any variant color is supported. Any VCF file can be uploaded. The files can come from RAID, but they can also come from your own pipeline. So there's no limitation on input as long as the files are in VCF format. And the input to AIM, again, is VCF format. We also support MAF or mutation annotation format. There's a question about trio analysis. We didn't show that example today, but you can upload a trio and then apply inheritance filters to the trio. That capability is available in Ames. I see a question about command line access. We do have command line access available. So you've been seeing the UI today. We do have command line access available, and we'll be expanding that capability in the future as well. So some of our databases, there's a question about the commercial databases. Anoop, when she was giving the overview of the databases, mentioned that some of these are commercial and some of them are publicly available. For the commercial ones, primarily the bio-based databases, so that's the human gene mutation database, the pharmacogenomic mutation database that we used in the demonstration today, TransFac for transcription factor binding sites, and Proteome for post-translational modifications. To get access to those, you'd have to acquire your own license from BioBase, but then we would deal with having those databases integrating into the software automatically. In terms of number of samples, there's a question about the limit of the number of samples that AIM can handle. There is no hard limit right now. It'll obviously depend on your deployment and the infrastructure that's available to the software, but there is no hard limit to the number of samples. I see a question about uploading users' own databases. So that's something that we're working on adding as a feature. So we're working to add a feature such that a user could upload their own databases themselves. We're also working on a feature to allow users to add annotations, custom annotations through the UI. And so both those features will be available this year. There's a question about gene annotation, where the gene annotations come from. So the definition of the gene itself comes from RefSeq. We also have ensemble gene annotations available in the software. In terms of annotation, so a number of the databases also provide information about genes, the disease databases, the cancer databases. So there are annotations about genes available from those databases as well. There's a question about the account renewal. Yes, currently we're on a subscription model, so you would have to, you'd have a yearly subscription to the software. If you've asked a question and you haven't heard it answered, if you can just type the question again in the Q&A box, you might have missed a few questions. Just ask your question again and we'll be sure to answer it. Yes, there's a question about Clinvar annotations. And Clinvar annotations are indeed included in the software. Let me show you that. On the product. And I can select either clinical significance or particular disease to filter variants on. Okay, I heard that we were muted and we might have missed some of that. So basically here I'm just showing an example of creating a custom filter using the Clinvar database. I've selected Clinvar out of the list of available databases. I've selected one of the annotations available from Clinvar, it's disease. I've selected a typo operator that we'd like to use to create this filter, and I can enter in any value here for disease. There's a question here about the protein domains that were in that gene summary plot. Those protein domains come from PFAM. I didn't mention export. There's a question here about exporting variants. So on the UI, you might have seen a place where there was text about exporting 1,000 variants. You can't, so the 1,000 variant limit is something we keep just for the evaluation or the demo version of the software. In your own version of the software, you could export as many variants as you like. The export formats are currently CSV, or comma-separated annotations, or MAS format, and we will be adding VCF export as well. There's a question about showing that the steps taken to get to the BRAF gene again, so I'll show that quickly one more time. So to get to that view of the BRAF gene, we started by creating some filters on a single sample level. We then applied them to all samples. We switched to the gene panel view. This is to study now a particular set of genes of interest in more detail. In this gene panel view now, any of these genes here in blue on the left side are linked to that detailed gene view that I showed for BRAF. So if I click on the gene, this takes me to the detailed view of the gene. And here we walk through what this plot is showing. There are multiple isoforms, 4 genes with multiple isoforms. There'll be a drop down here, and you can switch between isoforms to pull up this plot created against different isoforms. The summary table, again, is showing you information about each of the variants shown in the plot. The table below can be filtered using the plot above by clicking on any of the sites, or can be filtered using the drop-down here to filter down the table, either to variants just in the particular isoform you're studying, or just the coding variants in the particular isoform that you're studying. There was a question about the cancer germline filter. I'll open that up to show how that's defined. The default cancer germline filter includes genes defined in cancer gene census as germline, genes in HGMD, the Human Gene Mutation Database, genes in the GWAS catalog. But again, any of these filters can be customized. So you can select whichever databases you think is the most reliable set of genes to make this definition. There's also a gene panel filter available. So you can here enter in any list of genes and use that as another filter as well. So there's a question about the clinically actionable gene panel and the pharmacogenomic filter that gave us, that brought us to P53. So the pharmacogenomic filter we made just included all variants that are in the pharmacogenomic mutation database. And this database includes any variant that's been associated with either drug response or toxicity in the past. So that was a simple little filter we made to pull in all of those variants. The clinically actionable gene panel is a panel of genes that I created here just to have in my account. That's just a list of genes that have been previously shown to be clinically actionable in certain cases. So one is a gene panel, and one is a filter on all the variants in a particular database. There's a question about starting from a gene and then applying filters. So you can definitely specify a gene panel here. So you can filter your variants first for a set of genes and then continue to apply other filters on top of that. There's a question about that it looked like there were no Clinvar disease-associated genes in our NCI60 data. So actually, to pull in all Clinvar genes, we create a filter. And then include everything in Clinvar. And we should certainly see variants in these samples from Clinvar. There's a question about whether this software is part of our bioinformatics pipeline or if the subscription is at a private cost. The software itself has a subscription fee. If you're interested in both AIM and RAVE, there can be discussions about, you can definitely get access to both together, but AIM itself does have a subscription cost. There's a question about whether the samples have to be cancer specific, or would the user be able to upload VCF files from any tissue source? So the samples can come from anywhere. It doesn't matter, the example here is for cancer. It could be for any disease, anything that you'd like to study. There's no limitation there, as long as the file is in VCF format. There's a question about integrating with Bardic, which is one of the Somatic variant calling tools that's available in our RAVE software. So definitely any of the VCF files produced by our secondary analysis pipeline can be uploaded and filtered and visualized in Ames, including Bardic. There's a question about the secondary analysis software that we have. That's called RAVE, and RAVE stands for Read, Alignment, Variant Calling, and Expression. So it's RAVE. And there will be further webinars this summer that give more details about the RAVE product. And if you go to the website, you'll see more details about that product as well if you're interested in learning more. There's a question about which annotation sources are included in the subscription. So all publicly available sources are included in the subscription by default. The only one that requires your own license right now are the bio-based databases. That's HGMD, PGMD, Transfact, and Proteome. Cosmic as well has moved to a subscription model. So in that case, currently what we have in the evaluation version of the software is the free version of Cosmic. If you would like to use the most recent version of Cosmic, you'd have to get your own license for that as well. There's a question about pathway analysis. So that's not a functionality that we have right now, but that is something that we're working on adding to the software.
So there's a question about the recording of the presentation. We will email out, send you an email so that you can access the recording.
There's a question about how samples are uploaded, and I didn't show that here, so we jumped into the querying and didn't show how the samples are uploaded to the system. So I'll walk through that very quickly. So this is a list of data sets. Data sets are collections of VCF files that have been uploaded and after processing are stored on the data page. Once a sample or data set's been processed, it's available to integrate into projects. And then once samples are in projects, they can be queried using the query interface that you saw today. So these are some examples of data sets that have already been uploaded. I can click on, for example, data set for the MCI60 panel. So this is the upload page. Here you can add dataset name, select a reference genome, add any tags and descriptions, and upload your VCF or MAF files. And again, VCF files can be either one per sample or can be a single VCF file that includes all your samples together. After the VCF files are uploaded, move on to processing. Here you can select which samples to annotate. And if you have a trio, you can select which sample is the mother, the father, the proband, which, if any of the parents are affected, and the sex of the proband. Next, you can select which databases or resources to use for annotation, and you can either choose to select all resources, or you can choose one of our predefined annotation profiles. And at this stage, you initiate processing. Processing, again, is where your samples are actually annotated against the databases that you've selected. After processing, samples are available to bring into projects and move on to the querying stage. There's a question about the limit of the amount of data you can upload. Again, there is no hard limit. There's a question about extra storage costs, and yes, you would be responsible for storage of the data itself. There's a question about uploading raw sequencing data to AIM. You can upload your raw sequencing data to our RAISE product. RAISE includes tools for alignment, variant calling, so that's the whole secondary analysis process. After the sample has gone through RAISE, then you can upload VCF to AIM. But the product you saw today doesn't take raw sequencing data directly.
Okay, thank you, Aparna. And with that, we'd like to conclude the webinar. Feel free to go to our website, click on Test Drive Bina, or give us a call, and we can follow up with any questions that you come up with later. Okay, thank you very much.